Hello, my name is Julia Camargo and I'm an undergrad student at the Federal University of Rio Grande. And today I'll be presenting my research titled Analysis and Experimental Replication of Bone Artifacts from the Central Coast of Rio Grande do Sul, Brazil. So, as the name suggests, this research focuses on the understanding of objects made out of bones and antlers found in 4 archaeological sites that are located closely to one another. It analyzes the different types of artifacts and their manufacturing process with the aim of defining the chaîne opératoire behind it. This is an ongoing work, so all of these are partial results. The main hypothesis is that given the proximity and likenesses in between the sites, the 4 of them would have the same or at least similar technology when it comes down to bone-made artifacts. To start this presentation, I would like to tell you a little bit about the archaeological sites and the places where they are found. 3 of these sites, Ondina Brasil, Negrinho de Oliveira, and Newton Dutra, are cerritos, mounds usually found in marshy environments with great morphological complexity, varying in shape, size, and height. They are spread across the south of Brazil, eastern Uruguay, and Argentina. The construction of these sites is attributed to the Charrua, Minuano, and Guarani groups. The 4th site classifies as a shell mound. They are monumental man-made heaps of shells. But alongside the 2 shell mounds that stand out in the landscape of the Casca site, it is possible to identify a 3rd mound that stratigraphically is more similar to a cerrito. Casca and Negrinho de Oliveira have been dated approximately 2,000 years before present. These dates have not yet been published. As you can see here, these sites are located in between the ocean and the Patos Lagoon. The region's geological formation resulted from ancient sea level changes, creating a diverse system of marine barriers, sandbanks, lagoons, marshes, beaches, and dunes. This environment serves as a home to a vast variety of animal species. So here's how I've been classifying the artifacts. There are 3 main categories: perforated, perforating tools, and others. These categories divide themselves in types. To make this division, initially I considered morphology, raw material, and size. So as you can see here, there are 2 types of perforated artifacts, 7 types of perforating tools, and up until now only 1 type in the others category. Very briefly, here is one specimen of each type and their characteristics. Starting with perforated. Both perforated types are made in fish bone. Type 1 in fish vertebra, whereas type 2 is made from a hyperostic pterygophore. In the perforating tools category, the types were created according to the order I was identifying them. I believe types 1 and 2 are most likely the same type of artifact. They are separated because all the artifacts in type 1 are broken basically in the middle, but they're both made from metapodial bones and have similar shapes and measurements. Type 3 has 2 apexes but is made out of antler instead, and type 4 is also made from a deer metapodium, but it only has 1 apex. Type 5 is this one that is smaller and more arrow-shaped. Also, this bottom part is cylindrical. Type 6 is very similar to type 2, but lengthwise it has about half its size. Type 7 is very similar to type 1, but instead of metapodium is made out of another type of long bone. The others category is actually only 1 artifact, is this antler that has been modified. Now let's discuss the experimental part of this research. While I'm still in the early stages of this phase, experimental archaeology has always been fundamental to the development of this paper. For now, the experiments only have focused on perforating tools made out of bone. While analyzing the archaeological sites, I found 2 artifacts that provided a starting point for understanding how to replicate them. The first one being this epiphysis, which has been cut, and the second being this type 4 perforating tool that not only has a cut-off epiphysis, but also has a bipartisan diaphysis. Observing the rest of the material, I realized that there were a lot of items with cut marks on them, which led me to believe that this should be my very first step when dealing with experimentation. So instead of obtaining the piece of bone I wanted by fracturation, as it is observed on other bone industries, perhaps I should start by section. The second part of this process is to understand the resources they had available and what sort of tools could they be using to manufacture these things. Previous studies on the central coast of Rio Grande do Sul mentioned that lithics are normally sparse in archaeological registry. That is because, as we can see on the map, the whole region is mostly made out of dune fields with not a lot of stone available. In the literature, we can also find mention of mostly basalt sandstone, quartz, or polished granite. If we look at the lithostratigraphic map, we can find a few possible sources for this material, such as the basalt in the northernmost part of the state, the sandstone closer to the center, quartz and granite that are also not that far. In most of the sites that are part of my research, there are mostly very few very small agate and quartz flakes. All of this was taken into consideration before experimentation started. Another thing to be considered is that given that deer bones are not easy to come by in Brazil, they were replaced by sheep metapodials, which tend to have the same thickness, similar shape, but smaller size. There is a market for the consumption of sheep bones in handmade cutlery. However, they are normally treated with chemicals, mainly to clean, allow part of the fat on the bone to remain, and to prevent mold from growing. Even though the fat is preserved, these are dry bones that have undergone chemical treatment, so the results of the fractures when napping, when compared to the use of fresh bones, may vary. Cutting them, in my opinion, is less of a problem, but still dry bones are hard to work with, so they were rehydrated before the experiments, by leaving them in water for at least 20 hours before starting, and also by having water nearby to wet them during the experiment. My very first attempt that I will be describing here was to create a Type II artifact using sandstone to cut and to polish, given that that was a material I had abundantly in hand at that moment. So first, the debitage process. Both epiphyses are cut off, sectioning the bone, as shown here on the first picture. Then, to divide the diaphyses in two by partitioning them. I did one side at a time because cutting from the top both sides at the same time did not work. I couldn't control the flake as I wanted. So after dividing them, I started the façonnage, polishing using a sandstone block, focusing on abrasive moments on the ends, one at a time, creating the apexes. To finish the artifact, I thinned it a little bit by pressing the whole artifact against the sandstone and rubbing from side to side, so it could resemble the archaeological ones that are thinner, and also to keep the bipartitioned sides the same size. The last step was to round its edges a little bit, as seen here in the final result. Throughout the experiment, I took detailed notes and photographs, used technical sheets proposed by Mingatho's thesis. I tracked time, tools, stages, along with my own drafts to record every step. These personal sketches, while not artistic masterpieces, proved invaluable in documenting the specifics of my work. They helped me record what worked well, what mistakes I've made, and how I corrected them. My body and hand positions, the placement of tools I used, which parts were challenging, and the directions and movements involved in polishing and sectioning. I documented as many details as possible. The other attempts I've made at replicating the bone tools went much or less the same way. This, for example, is a Type IV perforating tool that I've replicated using agate flakes to section and this big block of granite to polish. Another experiment I made, even though cutting the bones has worked perfectly well, is fracturating them, with the only goal of observing how they break, and to compare that to what was left in the archaeological record. As I said before, experimental archaeology has helped me a lot with this research, even if it's on its initial state, because this was the very first time I had contact with this type of material, so in the beginning I was a little bit lost about what should I look for in the first place. So the procedure of creating these pieces has helped me raise questions about my sample. I've looked at what was left, those things that are not artifacts but are in the archaeological record, and questioned if these broken bones are broken because of taphonomy reasons or if they are broken with a method and a purpose behind. I am able to look at the marks in them, their textures, and see if there's something more in there that I should be paying attention to. So analyzing and replicating this material simultaneously has opened my eyes and expanded my knowledge, my goals, and my sample, allowing for a more complete work. Another thing in my mind when looking at this material is the resources. As we saw, there is not a lot of lithic material in these sites. perhaps because people would simply not leave their stone tools behind. But still, I wanted to look at other possibilities. So, thinking about this place full of sand, water, and shells, I've decided that I would try to experiment using something besides stones. I still have not tried, but I know that using sand to polish bone is possible. So I thought, what about the shells? So I've used Amianthus purpurata, common in the archaeological record, to cut and scrape the leftovers of my experiments. First with a normal entire shell, and then I broke it to see the difference. And it worked. The goal now is to use shells to fully replicate an artifact, reproducing this experiment in a more systematic way. Besides that, here are the very next steps on this research. To establish a replication protocol and the chain opératoire of other types of artifacts, especially those made out of antler and fishbone. To try different materials like quartz, basalt, and sand on the debutage and façonnage stages. To replicate all the types of artifacts already found, and to compare the experiments with archaeological artifacts to understand which of these tools was used in their manufacture, using a microscope to observe the markings left on them. This research aims to gain a deeper understanding of these artifacts, their stages of production, and the diverse ways in which these cultural groups interact with their environment and with each other and utilize its resources. Thus far, experimental archaeology has proven to be an indispensable framework for guiding this research, assisting in the validation of existing hypotheses like the debutage bisectioning and bipartitioning, and also the formulation of new ones. Before I end this presentation, I would like to thank the opportunity of presenting at this wonderful conference. I would also like to thank my advisor, Professor Dr. João Carlos Moreno, from the Laboratory for Evolutionary and Experimental Archaeology and Prehistory at the Federal University of Rio Grande. I'm especially grateful to Dr. Gabriela Mingatos from the Laboratory for Human Evolutionary Studies at the University of São Paulo, who has been co-advising me and guiding me through every step of this research. Without her support, this study would not have been possible. I extend my gratitude to the team at the Laboratory for Human Evolutionary Studies at the University of São Paulo, particularly Professor Dr. Mercedes Okumura, as well to the Interdisciplinary Laboratory for Research in Evolution, Culture, and Environment at the Museum of Archaeology and Anthropology of the University of São Paulo, and Professor Dr. Astolfo Araújo for welcoming me to their laboratories and enabling my experiments. Finally, I thank the National Council for Scientific and Technological Development, CNPq, for funding this research. Thank you.